Hi, this is Yue from School of Pharmacy, University of Pittsburgh. Thanks for being interested in our work. You might be familiar with immunotherapy and have been heard of its great performance in treating the cancer patients. However, a major problem of immunotherapy is only a small group of patients can respond to it, and what we are doing now is trying to make more patients benefit from immunotherapy. We are developing an integrative analysis method called Perturbed to identify the chemotherapy drugs who can improve the immunotherapy efficacy. The Perturbed utilizes the treatment-induced gene expression changes as the major feature to evaluate whether a chemo drug can induce significant improvement of immune response. The reason we use the treatment-induced expression changes, which is termed TAC here, is that we found comparing to the gene expression before and after treatment, TAC can best represent the difference between immunotherapy responder and a non-responder. We established the TAC signature for responders and non-responders. As you can see from the pathways, TAC signature for non-responder is enriched in cancer hallmarks, but the one for responders is very immune-related. We also look into the relationship between these tags with the immune cell infiltration, and we found the tags for non-responders are associated with immune cell exclusion, but the tags for responders are related to higher immune cell infiltration. Clearly, these two tag signatures describe the immuno difference between immunotherapy responders and non-responders. So next, we are curious about what will happen if we shut down the tags for non-responders in the tumor cells. Will the tumor cells become a immunotherapy responder? So here, we used the expression change intensity score to measure how much the tag signature will be affected by an shRNA or a drug. The amazing thing is, we found that if we shut down the genes from the non-responder tag signature, the tumor cells will have an increase on the genes in responder tag signature. This means the tag signatures are not only a phenotypic description of immunotherapy response. Instead, the tag for sensitive immunotherapy response can be turned on if we switch off the tag that make a patient resistant to immunotherapy. Therefore, we introduced the shift ability to measure how much a drug can switch the non-responder's tag off and turn on the responder's tag in a tumor. We performed an in silico screening on 5,000 drugs and compounds, measuring the treatment-induced gene expression changes and quantifying their shift ability. These are the top hits we identified, and they are the cases that have already been used in clinics to combine with immunotherapy for enhancing the treatment response. You can see the drug treatment will inhibit the non-responder's tag and at the same time promote the cell to express more responders' tag genes. The case I put here shows you the exact pathways that changed because of the drug treatment in multiple cancer cell lines. You can see there is a clear inhibition on cell proliferation, and meanwhile a stimulation on tumor intrinsic immune response in the tumor. Besides these reported cases, we also identified some novel synergy targets for chemo-immunotherapy combination. This PLK4 inhibitor is the one we are validating in our wet lab, and we have observed that a PLK4 inhibitor can induce a dose-dependent enhance of tumor antigen presentation, interferon signaling, and a PD-L1 expression. Together, our Perturbed, which is based on the treatment-induced gene expression changes, proposed a novel direction on reliably identifying chemo-immunotherapy combinations that can shift the immunotherapy resistance. Thanks for listening, and I will take any question.